Welcome everyone to our Integrating Bioinformatics Education Series. Our featured speaker is Dr. Sungwon Kim. Dr. Kim is a staff scientist at the National Center for Biotechnology Information. He is a computational chemist and cheminformatician, and he works on the PubChem project, which develops and maintains a chemical database called PubChem. And today, Dr. Kim will be talking to us about PubChem and its application for cheminformatics education. PubChem is a popular chemical information resource visited by millions of users every month. Dr. Kim will be providing a brief overview of cheminformatics and explaining some of the many data, tools, and services that come with PubChem and how they can be used to teach informatics skills for students. So, Dr. Kim, welcome and thanks very much for being our speaker today.
Okay, thank you very much for inviting me to give this talk. Okay, my name is Sung-Hwan Kim and I'm a staff scientist at the National Library of Medicine. And today I'm going to talk about PubChem as a resource for chemical information education. So here is the outline of my talk. First, I'll give you a brief overview of cheminformatics and then explain what is PubChem and what tools and services it provides. Then I'll talk about how PubChem can be used for undergraduate education with Cheminformatics OLCC as an example. So what is cheminformatics? Well, you may have seen some people say cheminformatics or chemoinformatics or even chemical informatics. Cheminformatics is more often used in the US and chemoinformatics is popular in some European countries, but all of these terms are basically saying the same thing, and they are used interchangeably. The term cheminformatics was introduced to the pharmaceutical industry in mid-1990s, and it first appeared in a scientific article in 1998. There are several definitions of cheminformatics, but this one is my favorite. According to this definition, it is a field of information technology that uses computers and computer programs to facilitate the collection, storage, analysis, and manipulation of large quantities of chemical data. And I added an emphasis on large quantities of chemical data. Basically, cheminformatics is about all tasks you want to do with a large set of chemical data. I believe many of you are from biology department or informatics or computer science department. So somehow you're familiar with bioinformatics. So it may be a good idea to think about this particular question before we move on. What makes cheminformatics different from bioinformatics? Well, there could be many different answers to this question. But in my view, the biggest difference is between cheminformatics and bioinformatics comes from what they deal with. In bioinformatics, we deal with biomolecules like DNAs, RNAs, and proteins. These biomolecules are essentially sequences of repeating units, like 4 nucleotide bases in DNA and RNAs and 20 amino acids in proteins. But cheminformatics deals with chemicals, and these chemicals do not have repeating units nor sequences. This difference is very important because it affects how to represent the structure of molecules and how to compare them with each other. For example, you can represent the entire human genome using a sequence of just 4 letters of A, T, G, and C. but that kind of simplicity cannot be expected in cheminformatics. For example, if you want to search a database for a particular protein, you just to go to the database and provide the sequence of that, provide the sequence in the search box, probably in FASTA format. Here, the sequence itself is, it well represents the structure of the protein. But let's suppose that you want to search for a chemical. What would you put in the search box? Because chemicals don't have repeating units nor sequences, so it is not very clear how to provide the input structure in the search box. Basically, we need a way to represent the chemical structure as a string of characters. So for this reason, many line notations for chemical structures have been developed, but the most commonly used ones are smiles and inches. I'm not going to explain the details about how to generate smiles and inches, but let me show some example. Here is the structure of Lipitor, cholesterol lowering drug, and this is it's a smile string. Here the equal signs represent double bonds, and the parentheses are for branches, and the @ signs indicate a stereochemistry of chiral atoms. And this is the inch string for Lipitor. It looks more complex than smiles, and the inch strings are not really human readable because its design focus was given to machine readability, not human readability. Just compare these smiles and inch strings with proteins or nucleotide sequences in FASTA format. You can see the impact of not having repeating units in molecules. This also affects how you compare molecules and quantify the similarity. Suppose that we want to compare similarity between proteins. For this purpose, we typically use sequence alignment algorithms like NCBI's BLAST. Basically, what these algorithms do is to find the best alignment between proteins that gives the best alignment score based on a substitution matrix like Blossom 62. But when you compare chemicals, we don't have repeating units, so we can't create a substitution matrix. So we need alternative methods for evaluation of molecular similarity. And examples are fingerprint-based similarity methods and 3D similarity methods. In 2D similarity methods, we use something called molecular fingerprints to encode chemical structures into a string of characters. This picture shows the concept of structural keys, which is a type of molecular fingerprint. Structural keys use a predefined fragment dictionary, and each bit position in a molecular fingerprint indicates the absence or presence of the corresponding fragment in the predefined dictionary. Then we quantify similarity between molecules by comparing their fingerprints. Basically, if there is a greater overlap between two fingerprints, then the molecules are considered to be similar. In 3D similarity methods, 3D structures of molecules are superimposed with each other to find the best overlap. Usually, 3D similarity methods are much more time-consuming, and but 2D and 3D similarity method search results often complement with each other. So here is a quick summary of what I said so far in bioinformatics. we deal with biopolymers, which can be represented as a sequence of repeating units. However, cheminformatics deals with chemicals, and they do not have sequences nor repeating units. This difference affects how to represent molecules and how to compare them. Now let's talk about PubChem. PubChem is a public chemical database at NIH. It contains information primarily on small molecules, but it also contains other types of chemicals such as siRNAs, microRNAs, carbohydrates, lipids, peptides, chemically modified macromolecules, etc. Essentially, what PubChem does is to collect chemical information from hundreds of data sources and disseminate it to the public free of charge. Currently, it contains more than 111 million unique chemical structures with cross-links to many other databases. And we provide various tools for search, analysis, download, and visualization. PubChem serves as a key chemical information resource for the scientific community in many areas, as well as the general public. PubChem is a very popular resource. For example, last October, PubChem had more than 4.8 million unique users. There are two things worth mentioning here. First, 4.8 million is a lower estimate. We use Google Analytics to track the web traffic, but Google has been losing the ability to do this in some countries, especially in Europe, because of the privacy laws and related regulations. So we have more users than what Google can track. And second, this number is only for interactive users who come to PubChem through web browsers like Google Chrome or Microsoft Edge. And it does not include programmatic users. And some users access PubChem programmatically through computer scripts, and the web traffic generated by these programmatic users are often comparable with the traffic from interactive users. Another interesting observation from PubChem's usage statistics is that PubChem is heavily used by young people. For example, in last October, about 38% of PubChem users were aged between 18 and 24. And those in this age group are likely to be college students, and many of them are required to take some chemistry courses like general chemistry and organic chemistry and biochemistry. One may ask how they use PubChem. Most people come to PubChem through Google. If you Google a simple chemical name like pentane here, you will see a PubChem record at the top or near the top of search results, and people click it. So there are 20 million college students in the US alone, and many of them take chemistry courses. And it is very likely that they will use Google to find some information on chemicals and click one of the top search results, and which happens to be a PubChem record. That's the reason why PubChem can have millions of users. So based on what I said so far, there are three big advantages of PubChem as an education resource. First, it is already being used by many young people. Second, it has been available to the public for more than a decade, and it's not going to go away in the foreseeable future. And third, it does not cost any money to students. Considering these advantages, PubChem has a great potential as an education resource for chemical information training. especially for small organizations like primarily undergraduate institutions and community colleges, which do not have enough budget for subscription-based resources like SciFinder and Reassist. Then what about large universities with big money? They are usually rich enough to buy the subscription for proprietary databases, but these resources are primarily for research, and it is not really convenient to access from outside of the campus. Importantly, students will lose access to these commercial resources when they graduate. So most students who will pursue non-academic careers after graduation will eventually rely on public resources freely available to them. So it is necessary to provide them with training opportunities about these public resources while they're in school. Now let me introduce you to some PubChem tools and services. There are many things that students can do with PubChem, but in the interest of time, I will cover only a few examples here. And one thing that I want to emphasize here is that everything you do with PubChem can be automated using a computer script written in common programming languages like Python or R. So average users search PubChem just for a single chemical, but there are people who programmatically access PubChem to get data for hundreds of thousands of chemicals, primarily for research purposes. Search by chemical name. Here is the PubChem homepage. And at the simplest level, you can search PubChem using a text query like chemical names. And let's suppose that we are interested in penicillin. So type penicillin in the search box and hit the enter. Then PubChem returns relevant records from multiple collections, including compounds, genes, pathways, proteins, bioassays, literature, and patents. And if we click penicillin record at the top, now we are directed to the compound summary page of penicillin. The summary page of a chemical provides a comprehensive view of all information available in PubChem for that chemical. Sometimes it's very long. So you may want to jump to a particular section that you're interested in by using the table of contents right column. For example, if you want to find molecular weights or hydrogen bond donor and acceptor counts, you click chemical properties and chemical and physical properties section. And if you want information on metabolism or mechanism of actions of the chemical, you may need to click the pharmacology and biochemistry section from the table of contents. Then search by chemical structure. There are so many things that you can do with PubChem beyond a simple chemical name search. For example, you can search PubChem using a chemical structure as an input. You can provide the input structure using a SMILES or inch string, which I talked about at the beginning of this talk. And alternatively, you can draw the input structure by clicking the Draw Structure button here and launching the PubChem Sketcher. And when you're done with drawing, you hit the search button. Then PubChem will perform various types of structure searches, including identity, similarity, substructure, and superstructure searches. And I mentioned similarity searches as similarity searches at the beginning of this talk, and substructure and superstructure searches are basically scaffold searching. And then a search by gene, protein, or pathway name. Sometimes your input query is not a chemical, but something else. For example, let's suppose that we have a gene or protein, and we want to find all chemicals that can modulate the activity of that gene or protein. Let's say we want to find chemicals or drugs that target the human acetylcholinesterase, also known as ACE2, which the coronavirus uses when it hooks into and infects human cells. So we type ACE2 in the search box and hit the enter. Then we get hits from multiple collections, and we click the human ACE2 gene here. Then you will reach the target page of the human ACE2 gene. The target page provides a target-centric view of PubChem data. Because we are interested in the chemicals that target ACE2, we click Chemicals and Bioactivities section from the table of contents. Now we have a list of about 300 compounds tested against ACE2. And if you scroll down further this page, you see two drugs for this target, and they are chloroquine and hydrochloroquine, which have been talked about a lot these days. And this target page provides a lot of information about ACE2. For example, if you go to the literature section, you will find a list of diseases that are commonly mentioned with ACE2 in scientific papers. And we see COVID-19 and SARS at the top of this list, along with the papers that co-mention ACE2 and the two diseases. Then PubChem periodic table and element pages. PubChem also provides an interactive periodic table of chemical elements. And if you go to the PubChem homepage and click the periodic table button, you see the interactive periodic table. If you click any element, for example, beryllium, you will see the element card for beryllium that shows some important properties like standard state or atomic mass and so on. And if you click this beryllium element page button, now you will get to the element page for beryllium, which contains much more detailed information. Now we are again on the PubChem periodic table page, and if you click the Game tab here, you can play this periodic table game. This was added as an educational feature to help test students' knowledge of element names and symbols. And one of the important goals of the PubChem periodic table and element pages is to provide elemental data in a machine-readable format. For example, if you go to the PubChem periodic table page and click the download button, you can download the elemental data and load them into a spreadsheet program like Excel or Google Sheet or computer scripts written in Python and R. So using these machine-readable elemental data, you can develop some useful learning activities like the one shown in this slide. You can ask students to go to PubChem, download the data, and to generate a chart for the ionization potential of elements. and explain the observed periodicity of the ionization potential in terms of electron configuration. And programmatic access. PubChem's web interfaces are optimized to perform very simple tasks commonly requested by the moderate users. But it is important to realize that everything you do with PubChem through a web browser can be automated using PubChem's programmatic interfaces. In addition, the programmatic interfaces allow you to do much more complicated and specialized things beyond what the web interfaces support. Importantly, programming skills are essential for automating routine tasks and processing and analyzing a large data set. So they're important for students pursuing STEM careers in the age of big data. PubChem provides multiple programmatic access routes, but PubRest and PubView are the most useful and the easiest to learn. And I'm not going into the details of them today, but my colleague and I developed a set of Jupyter notebooks containing sample codes in Python and R to teach students how to programmatically access PubChem data for further analysis and processing. And PubChem has been working with the chemical education community on a collaborative teaching project called the Cheminformatics OLCC, and let me talk about it for next couple minutes. Teaching cheminformatics for undergraduate chemistry majors has several unique challenges. First, cheminformatics is not a field of chemistry, but a mix of many areas, including chemistry, informatics, computer science, library science, pharmacology, biology, and medicine, and many others. Because of that, there are not so many faculty members with cheminformatics expertise at chemistry department at most schools. Second, there are no textbooks suitable for undergraduate chemistry majors. While several textbooks are available, they're more appropriate for those who already have some informatics backgrounds, like computer science or informatics majors with programming skills, or graduate students who are working on cheminformatics research projects. Third, cheminformatics is considered as a research technique, primarily for industry application, but not as a core chemistry skill set. And the Cheminformatics OLCC addresses some of these issues. In a sense, the Cheminformatics OLCC is a collaborative teaching project that involves instructors at multiple schools and external cheminformatics experts recruited from industry, government, and academia. This project aims to teach cheminformatics for undergraduate students and to develop free online textbook materials that can be shared with education community. In the Cheminformatics OLCC, excuse me. Okay. Informatics OLCC, the cheminformatics experts prepared online reading materials and problem sets for homework, and these course materials were made available online through the course website. Then the instructors at multiple participating schools used these online materials to deliver the cheminformatics course at individual schools simultaneously. And the instructors had face-to-face meetings with their students in the classroom. And at the same time, the cheminformatics experts got engaged in online discussion with students and instructors through the course website. So far, we have run this course 3 times, and here is the enrollment statistics. As you can see, we had somewhere between 20 and 50 students from 4 to 9 schools. And one thing noteworthy here is that in spring 2017, Central College had no student, but the course was taken by 3 faculty and staff members who participated in a faculty learning circle. And IQS is a school in Spain, and the course was offered as a non-credit seminar. So we had some flexibility in the way the course was offered. And all course materials developed during the Cheminformatics OSCC are available at the CCCE website, Committee on Computers in Chemical Education, and LibreText. LibreText is a free online textbook website. So if anybody wants to use these materials, they can use them for free. And here is the content map for the topics covered in the Cheminformatics OSCC. And some topics were covered in all 3 OSCCs, and they're indicated as white ovals. And many of these topics are related to PubChem and its data, tools, and services. Here is a quick summary of PubChem-related topics covered in the Cheminformatics OSCC that includes chemical information literacy and line notations like smiles and inches, and search by chemical name, search by chemical structure, and structure clustering, and structure activity relationship analysis, and programmatic access. And for the 2019 OSCC, we developed Python Jupyter notebooks to teach how to access PubChem data programmatically and how to perform some cheminformatics tasks using open source software packages, and how to use PubChem's bioactivity data to build machine learning models for bioactivity prediction of small molecules. And similar materials were developed for the R languages. And students had two options to run these notebooks. First, they could download and run them on their own computers. But in this case, some students have Macs and others have Windows PCs, and they have different versions of software. So they're likely to work with different computing environments, making it difficult for instructors to help them when they encounter any problems. So as an alternative, students were given access to a Jupyter Hub available through LibreText. In this case, all students used the same computing environment, so it's much easier for instructors as well as students. But this shared resource can be available to the students after the course is over, which is a disadvantage. And after the 2019 OSCC, Professor Joshua Schreier at Fordham University created mathematical scripts based on our Python notebooks and shared them with us. So we now have a sample codes in 3 languages, including Python, R, and Mathematica. And during the first two OLCCs, students were required to work on small project for the last 2 to 4 weeks of the semester. And they worked on a various projects ranging from Android app development to educational video tutorial creation. And a few students had opportunities to give a talk about their projects at a national meeting. And some student projects were evolved into more formal and long-term project, and one of them eventually became the basis of master's thesis. And as a side note, some students participated in the NIST Reference Data Challenge, and it was a competition for Android app development that used NIST standard reference data. And one of the students won the second place in this competition. But setting aside 4 weeks out of one semester for student projects made it difficult to cover a broad area of cheminformatics with the remaining time. So for the most recent OLCC, student projects were up to the discretion of the instructor at each school. And one school did student projects as a final exam. So we are going to have a next Cheminformatics OLCC in fall 21. And in addition, we will have another OLCC in spring 2022 about the Internet of Things. Actually, the OLCC's teaching model has been used to teach various topics since 1996, including environmental and industrial chemistry, pharmaceuticals, and chemical safety. And cheminformatics is just one of them. If you're interested in any of these two future OLCCs, please contact one of us. Okay, let me summarize this talk. Many PubChem users are likely to be college students, and PubChem has a strong potential as a resource for chemical information training because of its popularity, sustainability, and low cost. And PubChem supports various use cases beyond simple text search, including chemical structure search, search by gene, protein, or pathway name, interactive periodic table and machine-readable elemental data, and programmatic access. And PubChem has been working with the chemical education community to provide cheminformatics education for students. An example is the Cheminformatics OLCC, and it aims to teach cheminformatics for undergraduate chemistry majors and develop free online text materials that can be shared with the education community. And so if you're interested in collaborating with us, please reach out to us. I'd like to thank the PubChem team, LibreText, and CCCE members. And I also thank all people who participated in the Cheminformatics OLCC, with special thanks to Professor Belford and Professor Courts for leading this project. And I also thank NIH, NSF, Department of Education, and American Chemical Society for funding. And thank you for your attention, and I'm happy to answer any questions.
Sungwon, this is Bob. If nobody has a question, would you make a bit of a comment on data provenance within PubChem?
Well, okay. So let me put that in this way. PubChem doesn't have any well, how can I say? PubChem does not curate any information, but we do have curated information. So we are a data aggregator. So we collect information from here and there and merge them into one place. But when you have that kind of information from here and there, it is very difficult to realize who said what. So a PubChem, when you have this kind of heterogeneous data, you will see some discrepancies between data sources very often. So we PubChem has various PubChem is very interested in making clear how people how to figure out data provenance. That is, who said what, where the data come from. So when it comes to chemical information literacy training, checking the data source is the most important part of the chemical information literacy training.
Dr. Kim,
Yes.
were students interested in maybe getting more involved with PubChem? Are there any volunteer or other activities?
At the NIH or NCBI or
Yeah. So
yes.
just with the open training initiatives.
Well, really at the moment, well, there are several things for students. So NIH has a summer internship program. And so we provide a research internship for summer students every year. But this coronavirus outbreak basically, this year's summer internship was canceled, but we're going to have a kind of virtual internship next year. That's the plan. That's one thing. And another thing is that we used to have, before the coronavirus outbreak, we used to have something called a biohackathon. So we suggest some sort of small project that can be done within a short period of time with volunteers. We used to have like that kind of hackathon event like 3 or 4 times a year. But now at the moment they're kind of stopped. But I hope we will be able to run that kind of things soon. Yeah, and please reach out to us if you need any education need, because NIH is very interested in that area.
Well, if there isn't any other questions, let me ask you, Sungwon, could you very quickly describe the difference between a substance and a compound within the PubChem database? Because I think
Well, it's a kind of so as I mentioned previously, you know, PubChem collects data from here and there. So when you have information about some chemical, actually that can be from multiple places. So let's say, you know, some data source provide us methanol's spectral data, NMR data, and some data sources will provide us their stock information because it's chemical vendors, and some people provide a bioactivity of methanol against a particular target. So each of these information will be called a substance, because they're sort of an archive. So these individual pieces of information are stored in the substance database, and we organize them into a single record, and we call them as a compound. So simply put, when individual data depositors provide PubChem with chemical structures, we store them into a database called substance. And we extract unique structures from this substance database, then store them in a compound database. So yeah, so simply put, provided information which has redundancy, and compound is unique chemical structures without redundancy. Does that make sense to you?
Yeah, I thought that might be useful for people to know. Thank you.
Okay.